Hello, dear students. Myself, Dr. Akhilesh Kumar, Associate Professor of Botany, BCGC, W. Nagal Chaudhary. Today I will teach Golgi Apparatus. This is a kind of cell organelles which are present in the cell. Who discovered it? It is discovered by a very eminent scientist, Italian scientist, Camillo Golgi, cytologist as well as physician. He observed that when he stained the cell, he found that a stained nucleus of the cell. He described Golgi body as an irregular network of fibrils, small fiber-like structure present in the cell, vesicle-like cavities, and granules. While examining a nerve cell, a barn owl, he noticed that, like a networks inside the cells, and named them apparalo reticulari intimo, means internal reticular apparatus. Recognizing them to be unique cellular component, he presented his discovery before the Medical Surgical Society of Pavia in April 1898. In the 1950s, however, when the electron microscope came into use, the existence of Golgi apparatus was confirmed. Distribution of Golgi body into the cell. How it is distributed? Golgi body actually present in the cytoplasm of the most eukaryotic cell, and already we know that it is absent in prokaryotes and some mature mammalians, red blood cells, sperm cells of bryophytes and pteridophytes, and sieve tubes cells of plants. On the other hand, in liver cells, nerve cells, and various cells of the invertebrates and plants, more than one Golgi bodies are present that remain scattered in the cytoplasm. Animal cells tend to have fewer and larger Golgi body. Plant cells contain several small Golgi complex units known as dictyosomes. In plant cells, dictyosomes may make cellulose, and the vesicles associated with them may deliver the cellulose to be deposited in the cell walls. In deposited cellulose in the secondary cell wall, the vesicles that carry the cellulose must pass through the cell membrane. This suggests that they also play a role in cell membrane repair. Dictyosomes are stacks of flat membrane-bound cavities that is called the cisternae that together comprise the Golgi apparatus. Origin and multiplication of Golgi body. From where it originated actually? There are three different views have been proposed for origin of Golgi complex. First, endoplasmic reticulum. Second, nuclear membrane. Third, division of pre-existing dictyosomes. It has been proposed that the Golgi cisternae arise from the endoplasmic reticulum. The Golgi body remains connected with the endoplasmic reticulum, and it is believed to originate from the endoplasmic reticulum. Most likely, at first, a small membrane enclosed vesicles are formed by budding from the smooth endoplasmic reticulum. These vesicles then joined and fused to form the cisternae of the Golgi body. In the cells of some lower plants and animals, the Golgi body may be formed from the nuclear membrane and the cell membrane. Bauch, a scientist which in 1965 described the version of Golgi from outer membrane of the nuclear envelope in the brown algae. Vesicles are pinched off from outer nuclear membrane, which fuses to form cisternae on the forming face of dictyostome. It has been observed that during the cell division in both plants and animals, the number of Golgi bodies increases. In each daughter cell, just after division, the number of Golgi is almost equal to the nuclear in parent cell prior to division. On the basis of this observation, it can be presumed that dictyosomes also divide during cell division. Shape of Golgi complex or body. The shape of the Golgi complex is quite variable in different somatic cell types of animals. Even in the same cells, there are various in different functional stage. The shape is, however, constant with each cell type. It varies in form from a compact mass to dispense filamentous network. The Golgi complex is composed of flattened sacs known as cisternae. The sacs are stacked in a bent semicircular space. Cis entry face of the Golgi complex, which is a concave, lies near the transitional region of the rough endoplasmic reticulum and is usually oriented towards the nucleus, and its trans exit face is concave and is oriented toward plasma cell membrane. Just we can observe that the structure of the Golgi apparatus, it is made up of several stacks of parallel flattened sacs, cisternae, and may peripheral tubules and vesicles. In general, the Golgi apparatus is made up of approximately 3, 7, 8 cisternae in most animal cells and from 10 to 20 in plant cells. Although in some single-celled organisms, it may be consist of as many as 60 cisternae. They are made up of a stack of flat, circular, perforated membrane sacs containing lumen of 100 to 200 angstrom thickness. The number of membranous sacs vary from 10 to 20 with an interspace of 200 to 300 angstrom. The cisternae are equally spaced and separated from each other by thin layer of intercisternal cytoplasm or proteinaceous matrix. Actually, Golgi apparatus has a distinct polarity in both structure and function, having two faces, that is the cis and trans, for receiving and shipping departments. Just we can see that very clearly. The convex, that is the outer face of the stack, that forms the cis face, and the inner, that is the concave face of the stack, and that's during the maturing, this is called the trans phase. The whole of the Golgi apparatus is supported by cytoplasmic microtubules, that is the secretory materials reach the Golgi complex from smooth endoplasmic reticulum via transport vesicles which bud off from the smooth endoplasmic reticulum and fuse with Golgi cisternae on the cis phase. From the trans phase, secretory vesicles arise that carry the processed material to their destinations. Similarly, tubules, small round tubules arise from the periphery of the cisternae. Some of these enlarge at their end to form vesicles. Vesicles the lies near the ends and concave surface of the Golgi complex. These vesicles are two types: smooth or secretory vesicles and coated vesicles. All Golgi elements are filled with a fluid known as Golgi matrix. Cis Golgi network and trans Golgi network, how it works? Two networks, the cis Golgi network and trans Golgi network, are made up of outermost cisternae at the cis and trans phase. Vesicles containing proteins from the endoplasmic reticulum merge with cis Golgi network, allows proteins to enter Golgi complex. As CGN receives protein from the endoplasmic reticulum, it begins their modification, moving along membranes to membrane towards the TGN, that is a trans Golgi network. At other end of the Golgi complex, newly modified protein arrives at the TGN. From it is sent off to different parts of the cell via transport vesicles. These two networks are responsible for essential task of sorting proteins and lipids that are received at the cis phase or released at the trans phase by these organelles. The cis phase membranes are generally thinner than others. This is the structure of the Golgi apparatus. Just you can see that very clearly, which are very close to the endoplasmic reticulum, that is the cis phase, and after the maturing, just it is the trans phase which are responsible for the outgoing of the transported proteins. What are the chemical composition of the Golgi complex? Actually, it is a made up of phospholipids, proteins, carbohydrates, and vitamins. The phospholipids of this Golgi apparatus, that is the membrane and plasma membrane. Proteins and enzymes, Golgi complex from different plants and animals show great variation in the proteins. Enzymes, the main enzymes are glycotransferase, galactosyl transferase, adipase, CTPase, and NADPH, cytochrome C reductase. C. Carbohydrates, glucosamines, glucose, mannose, galactose, fructose, and glucosamine Plant cells like salicylic acid, xylose, arabinose are present in the plant cell only. Vitamins actually includes vitamin C and liberates it slowly into the cytoplasm in sufficient amount to prevent oxidation of the cell products. Just now we comes on the function, actually common function, not specific, common function of the Golgi body. Generally, Golgi apparatus perform the following important functions. Number one is that secretion, number two is the synthesis, number three is the apoptosis, number four is the phosphorylation, and number five is the sulfation. Just how this is secreted actually. Just you can see in the picture the endoplasmic reticulum, rough endoplasmic reticulums, they synthesize the proteins and goes to the cis phase and it get modified and transported and comes out from the Golgi apparatus to the trans phase, and they are transported by the vesicle into the outer across the plasma membrane. This path of secretion. Similarly, proteins which are to be secreted enter into the ER lumen while being synthesized on ribosomes attached to the endoplasmic reticulum surface. Transport vehicles bud from the SER carrying secretory protein which fuses with the cis Golgi apparatus. Already I have discussed. Now, just you can see that the actually secretion of the just and how it secretion of these proteins takes place and how it's the endoplasmic reticulum how this Golgi apparatus synthesized. Just nuclear membrane, nuclear pore, endoplasmic reticulum, smooth endoplasmic reticulum, ribosomes attached to rough endoplasmic reticulums, micromolecule, transport vesicles, Golgi apparatus, cis phase of Golgi apparatus, and transphase of the Golgi apparatus, and cisternae of the Golgi apparatus. These are the following steps followed for the transportation of the formed protein or modified protein from the endoplasmic reticulum. How this is the synthesis. Second point is synthesis. It is also a major site of the carbohydrate synthesis. Includes synthesis of the glycosaminoglycans. In Golgi apparatus, polysaccharides attach to the protein to form proteoglycans. Long unbranched polysaccharides are attached to proteins synthesized in the endoplasmic reticulum to produce proteoglycans, which are molecules present in the extracellular matrix of animals. Enzyme in the Golgi polymerize several of these glycosaminoglycans. Next function of this important function of these Golgi apparatus is the apoptosis, programmed cell death. The Golgi has a putative role in apoptosis, a newly characterized protein that is the Golgi anti-apoptotic protein almost exclusively resides in the Golgi and protects cells from the apoptosis by an unidentified mechanism. Just you can see that normal cell, it condensed and fragmentation starts with the help of enzyme, and this is the apoptotic dead of the body, dead of the cells. Phosphorylation, one of the important steps that are performed by the Golgi body. ATP is important into the lumen of the Golgi, utilized by resident kinases such as casein kinase 1 and casein kinase 2. Actually, apolipoprotein is phosphorylated in Golgi apparatus, which forms VLDL, consists of blood serum. The phosphorylation of these molecules help in their sorting for secretion into the serum. The next and last function of these Golgi apparatus is very important. Sulfation. Golgi involves the sulfation, adding of sulfo groups to certain molecules passing through its lumen via a important enzyme sulfotransferases that gain their sulfur molecules from a donor. The donor of sulfo group is the PAPS 3' phosphoadenosine 5' phosphosulfate. This process occurs on the GAGs, that is the glycosamine glycans of proteoglycans, as well as on the core protein. Sulfation is generally performed in trans-Golgi network. The level of sulfation is very important to the proteoglycans, signaling abilities as well as giving the proteoglycans its overall negative charge. This is the important function of the Golgi apparatus. Thank you.